Hello and welcome to NanoReach, a scientific outreach podcast by the Nanomedicine Research Group at the Institute of Chemical Technology, Mumbai. In this episode, I talk to Devashree, who is a PhD student at NRG. We'll get to know more about her journey and her research. Hello, Devashree. Welcome to the podcast. I'm so glad that you could make it.
Hi, Pat. Yeah, it's my pleasure to, do a podcast with you.
I wanted to start this conversation by asking you what made you pursue research in the first place.
Okay, so to give a brief background,
Mm-hmm.
I have done my MTech in biotechnology from DY Patil Pune,
Okay.
and so my dissertation was for whole year,
Mm-hmm.
and after that, for 1.5 year extra, I have worked in the same lab under Dr. Nilesh Kumar Sharma, and he's a brilliant scientist who knows how to sculpt a student's curious brain into
Mm-hmm.
making it more of making the science more fun
Wow.
to explore.
Mm-hmm.
So I think the whole credit goes to him because he planted the whole curious mind.
Mm.
It turned the whole curriculum into a very fun time.
Wow.
And working with him, he made us understand that how to find particular things which can be explored more, which can be known in detail,
Mm-hmm.
and that sort. So I think, yeah, that raised a lot of curiosity in me. The whole phase was very
So,
like mind blowing for me.
so you'd say that your experience as a master student and a JRF, it pushed you to pursue a PhD eventually.
Yes, absolutely.
Okay, nice. So, while I was preparing for this podcast, I went through your Google Scholar profile and I was so impressed by the amount of research you have done on cancer before joining NRG. So, I wanted to ask, like, what aspect of cancer research did you work on?
Okay, so the whole study was based on analyzing different, microenvironments in different cancers.
Mm-hmm.
Like it was a glioblastoma, it was breast cancer, it was liver cancer. So I have worked on multiple types of cancer cells, and I have checked on different microenvironment and how it affects the established cancer cells and their progression. So my dissertation was based on a glioblastoma drug, that is
Mm-hmm.
temozolomide, and its effect on a breast cancer cell. So that area was not explored much,
Okay.
and actually wanted to see how mechanism changes or how the cells react to it. So that was the basic funda that took ahead this research and ultimately led to number of publications.
Mm-hmm.
But the whole aim was to identify smaller things and focus on them so that you make up a good story out of it.
Yeah.
So in cancer per se, how a cancer adapts in itself in a human body, we
Mm.
give multiple toxic treatments to it, and yet finds a way to get away with the treatment. It still progresses.
Yeah, it develops a
Even
resistance towards the treatment.
exactly, even a single cell can ruin the whole physiology.
Yes.
So the whole mechanism is quite interesting.
Mm-hmm.
It is definitely deleterious for the human body, but it is very interesting to know how it does that.
Hmm. So, from a research perspective, it's fascinating.
Yes.
So, I wanted to ask, like, did you aim to do cancer research, or did it happen all of a sudden? Like, what was the story behind cancer research?
Okay, so in my masters, so my
Uh-huh.
master's course was an integrated course of a 5-year program
Okay.
in which it was BTech plus MTech.
Oh. Mhm.
And so I got one consolidated degree. And meanwhile, when my lectures were going on in the second year of my course, I had joined. I had wanted to help a professor of mine.
Mhm.
So she was working on completely a bacterial background.
Okay.
And initially, I started working with her so that with the thought that I learn some different skills, I learn some different ideas.
Mhm.
With that thought, I joined the microbiology field. And that was a different phase altogether. It was about learning how to isolate bacteria from hot water spring samples, and it was quite extensive.
Okay.
So the whole bacterial thought did not catch my mind in that, and I couldn't focus on that particular area
Okay.
that much. But I was always clear that I wanted to explore this cancer research field itself.
Mhm.
And during my dissertation I thought that no, I don't want to go in this area, so discontinued that project. And I started asking my professor Nilesh sir that whether he can accommodate me in his lab, and we can start working on some idea pertaining to cancer field.
Okay. Okay. So, moving on, like you did, I think 1 year of dissertation followed by 1 year of JRF.
Yes.
Okay. So, how did your journey at NRG begin? Like, how did you find out about the lab and what motivated you to do research here?
Okay, so one of my colleague who had recently joined Diva Patil as a faculty, she told me about. She had already passed from ICT,
Okay.
so she told me about Dr. Ratnesh, and his lab was working on some biological aspects. And I checked Ratnesh sir's profile, and it was very fascinating. The
Mhm.
way he was taking ahead this biologics field was
Yes.
it captures anyone's mind.
Hmm.
The curiosity which he also can develop, it's on a different level,
Yes.
and it was ICT.
Hmm.
So the reputation, recognition matters a lot.
Yes.
And I thought of approaching Ratnesh sir for the same.
Hmm.
And we both were very much fascinated by looking at each other's profiles. So because I had a number of publications, even
Hmm.
he thought of, yeah, let's give her a chance, which turned very lucky for me.
Yes.
And yeah, we got to know each other, and I am in his lab right now.
Hmm.
So the journey has been completely different than what it was in Diva Patil, in a very good way.
Okay.
So yeah, here
Yeah.
also we are exploring every scientific problem from a
Uh,
different perspective.
yeah. So for me, like when I was about to apply for the science communication role, I looked up on the Nanomedicine website,
Hmm.
and I found that there are diverse groups of researchers working on diverse area of research. And what I found was the common thing between them was they all were aimed at contributing somehow to the society, and that's what impressed me. So, what broad research area are you working at NRG?
Okay, so when I joined NRG,
Mm-hmm.
I was I was appointed on a project which was completely based on a bacterial field again.
Okay.
So I thought that I don't want to carry out the whole PhD work in a
Uh,
bacterial field.
yeah.
I wanted to jump back into the cancer field, which I always craved for.
Mm-hmm.
So I finished up a project based on probiotic research.
Okay.
And then, meanwhile, I was completely thinking about how to develop a cancer model, how to develop a device that can accommodate different type of cancers. We can
Mm-hmm.
study different mechanisms, drug interactions. I as and when I got time, I went through the literature. I thought of sculpting my work in the in this field itself, in the cancer research field itself.
Yes. Hmm.
So I was sure that, yeah, okay, cancer research पे तो काम करना ही है.
Mm-hmm.
So I started looking upon the different models which
Hmm.
lacks researchers' interest,
Hmm.
and I found that the retina is a particularly a very sensitive tissue,
Okay.
and there are a number of diseases associated with our retina.
Uh-huh.
There is a retinoblastoma cancer as well, which happens only in children below 5 years.
Oh.
So that also increases the complexity of this tissue.
Mm-hmm.
So yeah, my like I started drafting my research, work around this tissue itself altogether.
Okay.
So developing on different models of like pertaining to this particular retinal tissue.
Mm-hmm. Fine. So like that's how you transited from like cancer research, core cancer research, to
Yeah.
same cancer research, but in and
Organ
around
specific, you can say.
retina.
Yeah.
Okay. So now, like I know that you're working on developing an in vitro retinoblastoma model. So that means that you're developing a model in the laboratory that could be used to understand more about retinoblastoma, right?
Yes.
Okay. So how is this model different from the traditional models that are being used?
Okay. So retinoblastoma models are already there because there are established retinoblastoma cancer cells, okay,
Okay.
which are there in market. But what lacks in that is the ability to mimic the whole tissue. So
Hmm.
scientists have explored individual cells and the impact of drug on individual cells, but there
Okay.
is like the literature lacks the combination of combination of cell types and their effect on one another.
Hmm.
So there are, see, so in the whole physiology, what happens is there is a retinoblastoma tumor which is being developed, and underlying that are the several cellular layers which also get impacted.
Hmm. Hmm.
So in some cases, there are cases reported which has retinoblastoma metastasized into different type of cancer.
Okay.
See, after all, it's a cancer cell. It will, leach out and try to
Yes.
find a way to progress in completely different organ.
Hmm.
So I wanted to explore how we can develop a consolidated model where we can study the drug interaction, the effect of like cell to cell type, how they affect different cell types, how metastasis can occur. If we
Hmm.
can replicate that thing here, then probably we can look ahead into um, a larger picture would be to save animals that are used in preclinical studies.
Okay. Hmm.
So with that thought, we tried to develop a triple culture model for a retinoblastoma
Hmm.
uh, cancer.
Okay. So basically, you're developing this model to overcome the limitations of the traditional models. And one of the most important limitation is that the traditional models, they don't replicate the complexities of the cancer in humans, right?
Yes, yes.
Okay. So is this a 3D model that you're building?
Yes, you can call it a 3D model, but it's a static one. So what I mean by static is
Mm-hmm.
there is no continuous perfusion of culture medium.
Okay. Hmm.
So this is just I have used cell inserts
Mm-hmm.
onto which these cell lines are distributed in a different fashion.
Okay. So you're combining three cell lines?
Yes, it is a
Okay.
triple culture model.
Okay. Nice. Hmm.
So I have been testing different drug treatments and how to minimize the toxicity of drugs that are commonly being used by using some additional proteins,
Okay.
which can altogether lower the toxicity, because this is a cancer which happens in small children. Like
Hmm.
they are very young to face the whole
Side effects.
chemotherapy sessions,
Hmm.
and there are many more complications that occur even after they grow up.
Okay.
So to altogether to minimize the drug load, the
Mm-hmm.
toxicity which they have to bear,
Mm-hmm.
I'm trying to lower the concentration of the
Drug.
chemotherapeutic drug
Hmm.
along with some additional proteins.
Okay. Hmm. So are there any other projects that you're working on?
Yes, so my PhD topic has three different domains in it. One being the retinoblastoma, which we already discussed.
Mm-hmm.
Second one was development of a microfluidic device
Okay.
and developing a retinal culture over it.
Okay. So.
And so microfluidic device is a device where it's sort of a chip
Mm-hmm.
on which you have a continuous flow of media with
Mm-hmm.
growth nutrients, and so there's a polymer which is used to design this model, this
Okay.
microfluidic model, and in this model we develop different cell types.
Mm-hmm.
So the whole idea behind developing this was to generate a co-culture model that using two cell types which are
Hmm.
interdependent.
Okay.
So in this model, what we have done is we have used retinal epithelial cells
Hmm.
and retinal precursor cells. So precursor cells are a stage before they differentiate into photoreceptor cells.
Hmm.
So what happens is your epithelial layers acts as a feeder layer, acts as a base for the development of photoreceptors.
Yes.
And I have just tried to mimic that in this microfluidic device.
Okay. So, to give a perspective, like how big is this device?
To go into the dimensions of it, it is almost like 5 centimeter by 3 centimeter.
Oh,
Yeah,
and
it's a very small device. This model, it is very handy.
hmm,
Even the
exactly.
complexity, considering the complexity, the model is very handy, and you can real time, you can image it, you can analyze the growth of cells, you can tag them with fluorophores, and have a look at how it progresses, how it is developing.
Okay. So
So that,
basically, you're developing a retina on a chip, and which replicates the cellular functionality of a retina, right?
yes, yes, yes,
Okay.
absolutely right.
Hmm. So, in what way is this model advantageous over the other models, other traditional models, like consider animals?
The main significance of using a microfluidic approach here
Mm-hmm.
was to enable a continuous flow as it is being seen in the human body, human
Yes.
physiology.
Hmm.
So we are just trying to replicate some parameters that are similar to the in vivo conditions.
Hmm.
So we are trying to see if the gaseous exchange, if the exchange of nutrients, the growth factors, uh, you know, assessing the cytokine profile, if it is possible in this microfluidic device,
Mm-hmm.
and it can be replicated to a proper tissue which is, functioning
Hmm.
in a human body. So
Okay.
we are just trying to see whether if the cells are subjected to stress,
Hmm.
is it actually showing the effect as it is showing in a human body or not.
Oh,
So how exactly cells react to a specific
stress.
growth factor
Oh.
or
Mhm.
a stress which we give it.
Okay.
So all the parameters can be monitored on this smaller device. And what it will happen is, if we are able to mimic every parameter here,
Mm-hmm.
this can be scaled up, and many of the biopharma companies can, easily use it.
Hmm.
If say you have 10 drug molecules,
Okay.
so you can directly put an array of experiments where you have 10 different devices with respective controls in comparison,
Mm-hmm.
and you can just test your drug then and there.
Right.
So your time is saved, your cost is like cost for
Hmm.
building up every assembly is saved, and your resources are also saved.
Right.
So
Mm-hmm.
usually what happens is, in pharma companies, they use like they put a drug for a preclinical study
Hmm.
wherein animals are used to test these different type of drugs.
Mm-hmm.
So after the drug treatment, the animals have been sacrificed to understand what happens in their physiology. They are the particular organ in which the drug is subjected, or drug interaction has to be understood, has to be studied.
Mm-hmm.
The animal is sacrificed and the organ is studied.
Yes.
So what we can do is, we can just, like, this technology can eliminate this preclinical studies altogether
Hmm.
once it has been completely validated.
Yes. Right. And what's your third objective of the PhD project?
Okay, so third objective pertains to another retinal disease.
Okay.
So the first objective was retinoblastoma. It is already a diseased model. Okay,
Yes.
the second one was to develop a microfluidic model,
Mm-hmm.
wherein we use epithelial cells and the growth factors from that to make the retinal precursor cells differentiate into different type of photoreceptor cells.
Okay.
And the third one was regarding a disease called AMD, that is age-related macular degeneration.
Mm-hmm.
And this disease happens due to accumulated stress signals along the as and when the age increases above 50 or 60 years of age, a
Mm-hmm.
human body starts to deteriorate and the coping mechanism is also compromised.
Yeah.
So when the stress signals accumulate and usually they are flowing into your system, but the bigger organs might not respond to it that in like causing that much of a complexity.
Mm-hmm.
But these complexities are seen to be affected to in our retina. So what happens is, as and when these stress accumulates and targets a specific retinal tissue,
Mm-hmm.
here it is found that the vision is compromised, and this is a progressive disease.
Okay. So it could lead to blindness as well?
Yes, it leads to blindness, vision loss, because what happens is these so a photoreceptor layer is very sensitive.
Hmm.
So it is like even in day to day life, if you can see, after a whole stressful day, your eyes get stressed after looking at a screen
Right.
or at some bright light. Your
Hmm.
eyes get stressed at the end of the day.
Mm-hmm.
So you can feel that stress. So
Yes.
that is when your retina is trying to recalibrate itself and say that okay, we have to
Rest.
perform our uh, functions properly.
Mm-hmm.
And the next morning you're fine when you have the particular rest.
Yes. Yes. Yes.
So what happens is the stress signals, the light, be it lifestyle changes, these stress response, what it does is it goes and directly acts your photoreceptors layer,
Okay.
and they start disrupting this layer from the base layer, that is the epithelial layer.
Mm-hmm.
So then what happens, and photoreceptor cells, they have very um, lesser precursor cells to regenerate themselves.
Okay. So then they don't grow back as fast as
Yeah,
other
they
cells.
don't grow back. And at a later age, 60 or 65, 70, so at that time the body do not have enough cells to regenerate itself.
Yes. Mm.
So that is when your whole body starts aging. So when it starts aging, you don't have any reserved cell bank that will regenerate every cell of your tissue.
Right.
And once it has been compromised, it cannot be grown back easily.
Mm-hmm.
So even at that stage, the drug treatment, whichever doctors prescribe,
Mm-hmm.
might not be useful or might not help in regaining back the vision that has been compromised.
Mm.
So what we are trying to do is we are trying to replicate the same model,
Okay.
which is there for age-related macular degeneration, that is AMD. We are
Mm.
trying to develop the same diseased model so that we can have better prognostic and curative way for developing multiple drugs that can scavenge these stress signals.
Okay. Mm-hmm. So even is this a 3D model as well?
No, this it is a 3D model because it is a co-culture model, and there is development of different lipid layers between these two cell types.
Okay.
So yes, this is also a sort of a 3D model. It does not involve any scaffold based,
Ha ha ha.
but it is a 3D model. Like we can visualize it in a Z axis as well.
Oh, okay. That's great. Mm.
Yeah.
Okay, fine. So, like, so let me just summarize your research, right?
Yeah.
So basically, you're working on different eye disorders like retinoblastoma, age macular degeneration.
Age related macular degeneration,
Okay.
and the other one was the microfluidic device on which normal retina is being developed.
Hmm. So what I observe is, like through your research, you are developing better models that can potentially be used as alternatives to traditional models like animals. So why
Yes.
do you think it is important to have alternative to animals?
So animal models, what happens in animal model is
Mm-hmm.
sometimes they are not see I say in terms of retina.
Mm-hmm.
So animals like rats, mice, they do not replicate the whole macula which is there in the human eye.
Okay.
So they lack that macula over there.
Yes.
So even if we have to understand a physiology, like a drug, pharmacodynamics of a drug in
Mm-hmm.
animal, the the whole system will be not replicated in the human body as it is.
Mm-hmm.
They will show a particular effect on particular cells, but it might not be the same profile for a human eye.
Okay.
So there is some difference between using an animal model and actually implementing it in a human body.
Hmm.
So I think to avoid the whole animal like testing on animal model, what I feel is if we develop specific diseased models, be it microfluidic or be it static, microfluidic being more advantageous because it will be able to replicate more of a physiological parameter.
Yes. Hmm.
So, if we develop a microfluidic model of particular disease,
Mm-hmm.
so we can anytime use that and study all the parameters, study the distribution pattern of the drug. We can also attach different organ specific models to it to see what happens in the ADME, the absorption, and excretion, metabolism and excretion
Hmm.
mechanism of the drug. So we can create the whole human body, this whole distribution pattern of the drug as and when it enters into the body and it leaves the body.
Mm-hmm.
So we
So
can see it in a very orthogonal approach.
yes. So it can be
And
said, huh?
yeah, one more point I would
Mhm.
like to add is that in this particular in vitro model, we can also develop a personalized models which will allow us to focus on a personalized medicine or personalized therapy for that particular patient.
Okay. Mm-hmm.
So the some of the cells can be isolated, it can be cultured, it can be grown, and it can allow us to understand the mechanism for that particular patient and allow us to pick out like some drugs that will act more specifically towards that patient
Patient.
profile.
Hmm. Yes. Interesting. So it can be said that you're developing models which could minimize, animal usage by providing more accurate and predictable results, right?
Yes, yes.
Hmm. Yeah.
So
So,
trying the testing a drug on animal model, it is very time consuming. It requires multiple ethical consents,
hmm.
and after that, even some like there is a 90% chance that a drug might fail
Hmm.
in the whole drug development pipeline, and that is what happens. So the failure rate for any drug to enter into a clinical trial might decrease if
Yes.
this technology is used, or
Hmm.
you can have a preliminary understanding as well as and get a detailed mechanism out of it.
Yep. Hmm. So fine. Now, I think I'm done with the research related topics that I wanted to discuss with you.
Okay.
There are next few questions that I want to ask personally because someday I aspire to pursue a PhD too. Okay.
Okay.
So I wanted to ask importantly, how do you deal with setbacks or failures? I don't want to call it failures because it has a negative connotation to it. But
Mhm.
as a researcher, on a daily basis, we do face with experimental setbacks or certain setbacks in the laboratory. So, and everyone has a coping mechanism to it. So, what's your coping mechanism to experimental setbacks?
Talking about setbacks, I think thinking about it from a different angle really helps because see after all we are researchers,
Hmm.
we have that curious mind that if a experiment is failing, why it is failing, how
Yes.
it is failing.
Hmm.
So if we have tried it 100 times and we have failed for 99 times, we know what
Not
not
to
to
do.
do and one way how to do it.
Yeah.
Though that is just one way, it's not that significant, we need to replicate it many more times.
Hmm.
But we are very much sure that we can document a protocol based on how to avoid failures.
Right. Hmm.
So I think dealing with failures is a part and parcel, but We have a dynamic environment in our lab.
Okay.
So there are people working in different domains altogether.
Hmm.
One like me. I'm working in a cancer pertaining subject. Another one will be working on a protein characterization subject
Hmm.
domain. So it is a very wide array of like friends and student setup we have here.
Hmm.
So their insights help a lot in this setback, as you said now,
Hmm.
that if I fail, their aspect of looking at my failed experiment might be completely different.
Huh.
They can guide me, not not exactly how the experiment should work,
Hmm.
but they can address a problem from their different angle, and
Okay.
that angle helps us think, okay, yeah, this can also be a way to think about it.
Hmm.
So like considering um, different people's approach to look
Yep.
at that problem also helps a lot
Hmm.
in this way.
Right. That's nice
And
to hear. Huh.
uh, interacting, going out and clearing your mind does help in a field like ours.
Yes. Hmm. So, the next question is one of my favorite questions that I ask to researchers. So, the question is, what is an unexpected skill that you picked up during your research, but this skill has come in handy to you in your life outside a lab? So, in my case, during my master's, I was working on Drosophila to understand growth.
Hmm.
And uh, during this time, I used to cook a lot of fly food weekly, and the fly food has to be perfect in the sense that the cooking time has to be optimum, the quality and quantity of ingredients has to be perfect, or the fly food will be a waste. So cooking this meal regularly, it has helped me obtain or acquire cooking skills that I used because I used to stay away from family. So
Hmm.
do you have any such stories that you'd like to share?
I would just like to mention one point that
Mm-hmm.
the throughout my research field, what I have learned is every minute detail counts a lot. Like be it time, be it your experiments, be it anything.
Wow.
So what happened was in one of my experiments, just like working on this development of disease model objective,
Mm-hmm.
I happened to like subject a cholesterol based medium to my cells,
Okay.
and they formed a completely different morphology. They were not stressed, but they
Hmm.
formed completely different and beautiful extensions towards each other.
Okay.
So this was just a trial experiment, which I was like, yeah, let's see what happens.
Mhm.
And even though there were very few cells expressing those profiles,
Mhm.
but that was a topic which I had to explore more about it. I had read about it in the literature, but I couldn't find anything that is causing this difference.
Mm.
And going into this whole arena is completely mind blowing for me right now,
Yeah.
because like I may or may not find a very, like greater difference or a different pathway altogether,
Mhm.
but at least I'm satisfied that okay, this small change in my cell morphology is something to look into.
Yes.
And had I not tried this whole new set of experiment, I might not have explored that my cells can behave this way as
Mm.
well.
Yeah. This is a great skill to have because having such skills fuels discoveries, scientific discoveries.
Yes, and one more thing is, as and when you do your experiments or research, whatever.
Mhm.
So what happens is, you are doing a particular thing, be it Drosophil or be it handling cells in my condition.
Mhm.
But when we are doing it, we can actually picturize what can and what might happen. There is a whole pathway which you can imagine that okay, this growth factor might target this particular pathway. You can actually imagine the whole scenario in front of you,
Mhm.
and that has helped me in my research life as well as in my personal life.
Oh, That's great.
So in every situation, I picturize it that way. Like,
Mm-hmm.
be it any decision making, a smaller, bigger, any action which I have to take.
Wow.
So I can imagine it in that way, that, oh, okay, if this happens, then yes, no, then this, what action will it cause? So
Mm-hmm.
I think this has developed this additional personal skill which I have right now.
Nice. So, I understand that PhD is hectic and it's fun at the same time,
Sure.
but how do you manage to squeeze out time for your hobbies? First of all,
So,
tell me what are your hobbies?
okay, so I am a rider. I like to ride bikes.
Oh.
And
Mm-hmm.
second hobby I would say is I like to play badminton as well.
Mm-hmm.
Mm, yeah, I have developed it in like a year or so.
Wow.
And I like to read a lot.
Mm-hmm.
So hobbies can be defined in very different ways.
Yes.
So these are some of the primary things which I like to do. I like to draw, I like to dance. So there
Hmm.
are multiple things which we do.
Right.
But squeezing time, as you said,
Mm-hmm.
it is not easy to take out time from your daily schedule, because as you know that you have things in your mind that you need to complete.
Hmm.
And it becomes very difficult for you to discontinue the day when you know that, okay, this might be the result of it.
Hmm.
So your driving force to conduct any particular experiment might take up the whole day, take up even 24 hours of your whole day.
Yes.
But if at all, some this happens only in some days, but in most of the days what happens is you get back from the lab at 6, 7, 8, and
Hmm.
then you have this completely drained mind, and that is when your brain signals you that no, you have to go out and do something which you like.
Hmm.
And automatically you start making time for yourself,
Hmm.
because um, how much ever you love doing your research, doing exploring your scientific work, but coming out of it and leading a normal uh, doing normal activities, leading a
Yeah.
normal life is also
Important.
important.
Hmm. Exactly.
Yeah.
So, one final question before we leave. Okay.
Yeah.
So, do you have any advice for budding researchers?
So my advice would be,
Mm-hmm.
you should not have the fear of failures. It has happened so many times, like
Mm-hmm.
it has happened with me itself. Okay, so that is the learning,
Hmm.
that I am trying to analyze a protein, its expression, but I know it is there. But
Hmm.
even though it is there, it's not been expressed.
Hmm.
I don't know why, and I keep on doing it, keep on doing it, and one fine day, after conducting it for 14 or 15 times,
Hmm.
I got it. So I know that yeah, okay, failures are going to be there,
Hmm.
but you should not give up the thought that uh, just because you're going to fail, you're not going to go in that path. You're not going to
Hmm.
explore it. So the fear of failure will just drag you down. It will be just an
Yes.
anchor in your life. And nothing productive can come out of it.
Mm-hmm.
So yeah, and consistency. So in the starting of my PhD, it happens with everyone though.
Mm-hmm.
In the starting years of my PhD, I was completely filled with energy. Like, yeah, I have to do this, 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 this. Multiple things were planned throughout the day,
Mm-hmm.
and I used to accomplish those things. But then there was a time when I started experiencing, okay, this is not working out, that is not working out, and everything started to tumble down.
Okay.
I had a talk with my guide
Mm-hmm.
that I am feeling so helpless right now.
Mm-hmm.
And she, Dr. Prajakta Dandekar, she's
Mm-hmm.
my guide, and she said that don't worry, even if you don't get the results, it is okay,
Hmm.
but you keep working on it.
Mm-hmm.
So that consistency was somewhere lost, and it happens with everyone. But to regain back that zest or that vigor to perform your work is also very important.
Yes, 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 absolutely.
So there will be days where you feel very helpless, and you just quit and go. But after that also you have to come back, you have to gather energy to start again.
Hmm.
So I think that is the main advice which I would give, because it is very important for a PhD. PhD is a very big commitment
Yes,
altogether,
exactly.
and giving up in a very short notice just because you have experienced a like failures from every aspect doesn't mean you quit it.
Mm-hmm.
You have to pass through that whole phase where you prove to yourself and not to the world.
Yep. Wow. That's
So that,
that's a great advice.
yeah, thank you.
Okay. So, all right. This brings us to the end of our podcast. Thanks a lot, Devashree, for taking out time, especially on a weekend, and talking to me on this platform. Mhm.
Thank you, Parth. That was my pleasure to interact with you. And yes, I enjoyed a lot sharing my views.
Yes. So, all the
Yeah.
best for your
Thank you so much.
PhD.
Thank
Yep.
you so much, and wish you the same
Yes.
for
Thank you.
every aspect of your life, rather.
Thank you.
Yeah. So, bye. Thank
Yes.
you.
Bye bye.